Yeah.
Okay. So I'm going to just give some background and then I can go over figures. I mean, typically journal club is, I mean, I'm happy to talk about whatever you guys want to talk about, but I was feeling, first I'll give some background how we ended up writing this paper up. I mean,
Yeah.
this work, because yeah, anyhow, I'll start when you tell me to start.
Yeah, that would be great. So there are also like usually a lot of clinicians who attend.
Yes.
So, if you can just like maybe also point attention towards the translation
Oh,
meaning
definitely,
their work,
definitely,
that would be great.
definitely. Or reverse translation, as we call it sometimes.
Yeah.
Okay. I would say some of these observations may not be that, you know, surprising to a clinician actually working in the ketamine field per se, but we're trying to see it at the basic level, synaptic level, what they mean.
Yeah, it's fantastic.
Should we let people in? And we usually we give people sort of 3 minutes grace or
Yeah.
so forth.
And Rupert, you're hosting and you will then send
Okay.
the recording, right? Or have
Yeah.
you already
Okay. Yeah.
started the recording?
We're recording at the moment, so I'll just leave it running.
Okay.
Dr. Cavallali, the number of attendees is a little bit variable. Sometimes we have up to 30, but it can vary. And I think we'll give everybody 3 minutes or so. We usually give people 3 minutes grace and then crack on, because it is recorded, so we can people can always catch up if needed. And I think Sarah will introduce you, if that's okay.
I'll be very brief, so maybe then you can add whatever you deem is relevant.
Sure.
Why don't we crack on, Sarah?
Can you start? Yeah.
Yeah.
All right. So welcome back, everybody, to the Ketamine International Journal Club. It's our pleasure to have with us today Dr. Cavallali, who is the professor and chair of the Department of Psychopharmacology at Vanderbilt University. And as you can see, is going to talk to us about their work on ketamine-induced synaptic plasticity. Dr. Cavallali, over to you. Thank you.
Thank you very much, Sarah, for this invitation and covering this paper. So briefly, this is actually a paper by supervised by myself and Dr. Lisa Monteggia, whom I've been collaborating for a long time, which I'm going to get into some of the earlier work in a second. But the work is done by a graduate student, very talented graduate student, Michelle Piazza. And please feel free to interrupt me if there are any questions. I can probably see, you can shout out or you can raise a hand. Sometimes it gets awkward in these Zoom meetings how to actually see, but I would prefer to actually have an open discussion, which I think hopefully we're going to have at the end regardless. But the point is, so ketamine, as you all know, I don't think needs much introduction to this audience, that triggers a rapid antidepressant effect. And I'm not a clinician, I'm a basic scientist. I work on synaptic transmission, and this is something that me and Dr. Monteggia have been interested for a while. And she comes from more of a background of psychopharmacology, whereas I come from more a background of synaptic transmission, very basic mechanisms of synaptic transmission and synaptic plasticity. So we've been joining forces on this question because one of the things that actually attracted us that this is a tractable issue. So as you well know, classical antidepressants take several weeks to a month or so to show efficacy, and they normally target monoaminergic transmission, and their mechanism of action has been somewhat, let's say, ambiguous in a sense that, yes, they target monoaminergic transmission early on, but the effect takes such a long time that probably other processes such as chromatin remodeling, circuitry organizations, and neurogenesis and other processes, they've been all postulated to be involved. When it comes to ketamine, though, it's a game changer because it works really fast within hours and at very low doses. It shows efficacy in a vast number of patients with different types of depression. That, please feel free to correct me. I'm not a clinician, but that's what I've been over the years, been talking to clinicians about this. And it has been surprising that this works so fast because what has been thought is to actually really treat depression, you really have to take a fundamental way to alter the neurocircuitry or trigger neurogenesis, etc. Maybe you cannot actually treat it that fast. And this type of purely clinical work suggests that, this is actually can be treated extremely fast using drugs like ketamine. So, and ketamine, as I'm going to actually show more of the data rather than showing cartoons, it is an NMDA receptor blocker. And our work and several other studies suggest that it indeed works as an NMDA receptor blocker to trigger this effect. There have been other targets that have been proposed, but I'm more than happy to discuss further that point is that I think all the evidence suggests that it is actually, its action is an NMDA receptor blocker action that triggers this. Now, this already presents a puzzle because I believe you will all be familiar with the whole literature on Hebbian synaptic plasticities that kind of implements Hebb's original postulate of how you may actually encode information in the nervous system. And these Hebbian plasticities, the most famous one, of course, is the long-term potentiation. And that requires activation of NMDA receptors. So that is a form of synaptic plasticity that requires activation of NMDA receptors. And that NMDA receptor in that context is largely thought to be a coincidence detector, that the release of glutamate is sensed by the receptor, but it also requires postsynaptic depolarization, activity of the postsynaptic neuron, so that the two inputs, the input and the output, are both co-activated, and that results in synaptic strengthening. This is some very basic neurobiology, but this process has been studied intensely, and several mechanisms are, really identified and thought to be cellular synaptic correlate of memory. Now, on the other hand, what we have here is an NMDA receptor blocker that triggers some form of plasticity, which soon I'm going to get into explaining what that is. And this plasticity somehow alleviates symptoms of depression, but also largely does not interfere with cognitive processes. So patients can be treated, maybe some initial, bout of confusion, etc. This plasticity is triggered and is such that, the patients can actually function normally afterwards. So there is no significant impairment in memory. And this actually comes from the clinical work. And I'm more than happy to discuss. Probably there are experts in the audience more than I am on this thing. So we then embarked on this study to actually really kind of interrogate this specific question in a more, let's say, reduced setting, looking at brain slices, hippocampal brain slices, which we think hippocampus actually is a major trigger for the antidepressant effect. And coincidentally, it also the site of this memory, that LTP type phenomena. So let me see if I can try to move my, okay, why this doesn't, oh, okay, good. So this started to basically the question started with a paper, with a fancy paper we published a while ago, 2011. And in this paper, we proposed that an NMDA receptor blockade under resting conditions, a concept that I'm going to introduce in a second, by ketamine triggers the rapid antidepressant response. And one of the things that we could replicate in mice, ketamine's antidepressant response, using these, depression-related behavioral paradigms like forced swim test, novelty suppressed feeding test, etc. And in addition, what we could show, and this is one of the figures from this paper back in 2011, is that application of ketamine, if you see here at the below, for 30 minutes into a hippocampal slice at the CA3, CA1 region, actually causes a gradual potentiation of AMPA receptor-mediated responses. So ketamine blocks NMDA responses, but causes a potentiation of AMPA receptor-mediated responses. In a way, just the mirror opposite of LTP in a way. For LTP, you have to activate NMDA responses, NMDA channels, but that also results in a potentiation of AMPA receptor responses. So AMPA receptor is the key receptor that carries out fast glutamatergic transmission in the brain. And in this figure, there are a couple other, this is one of those papers where everything is packed into little figures and several supplementary figures. But the point is, there is this, which I'm not going to get into that much into the detail, but there is this elongation factor in the dendrites that seems to be dephosphorylated when you actually treat with these NMDA receptor blockers. And this dephosphorylation can occur even in the absence of activity. So activity is not required to trigger this plasticity. I mean, at the end, of course, activity stimulation is required to observe this plasticity. But to trigger the plasticity, you actually don't need activity. And now below here, you can see these resting NMDA receptor activities that are blocked by ketamine. So that basically suggests that, yes, ketamine is actually working to block NMDA receptor responses. And this is triggering plasticity. So in this study then, back in 2011, we come up with a model that how this might be working. What we suggested is that what is going on is this spontaneous neurotransmitter release, glutamate release, activates NMDA receptors, and that in turn activates this kinase, which is actually a calcium calmodulin-dependent kinase. It is a protein that phosphorylates other proteins, adds phosphate residues to other proteins, but it is activated by calcium signals. And it only has one known target. That's this elongation factor. And this elongation factor in its phosphorylated form suppresses protein translation. And a key factor here that needs to be translated is BDNF. That's brain-derived neurotrophic factor. And this factor is important to trigger antidepressant effect. What's interesting is in this study, what we showed is that this BDNF is critical for rapid antidepressant effects of ketamine, but it's also critical for what has been known before this work, for action of classical antidepressants like SSRIs, but in the more long term. So it seems like BDNF signaling is some sort of a convergence factor, point of convergence between the rapidly acting as well as more traditional antidepressants. And somehow these traditional rapidly acting antidepressants tend to cut to the chase and get to that target faster than SSRIs. So what happens is that if you put ketamine, you block this resting NMDA receptor activity, then you deactivate EF2 kinase. That results in dephosphorylation in EF2, and that kind of relieves this brake on BDNF translation and then activates TrkB. That leads to this AMPA receptor-mediated potentiation. And here is the data from a subsequent paper we published. And up here, it's a crowded figure, but the message is very simple. So you apply ketamine, the way I showed before. We replicate our own data. You can see potentiation. And it's interesting, of course, you would be worried that where you're putting an NMDA receptor blocker, is this actually having, blocking NMDA responses? It is blocking resting NMDA responses. But unless you continue to keep it during stimulation, it does not block evoke responses. Evoke responses are there, and they can be gradually blocked if you keep actually stimulating in the presence of ketamine. So what we're doing is we're only blocking these NMDA blocker receptors that are only active under resting conditions. So key thing about ketamine to remember, it is an open channel blocker. The channel has to be open to be blocked. If the channel is not open, it doesn't get blocked. So therefore, only the channels that are open under resting conditions get blocked. Now here you can see we can trigger this potentiation, and importantly, doesn't have much effect on the presynaptic side. These are some measures of presynaptic efficacy. It is more of a postsynaptic effect. And then MK801, another open channel blocker NMDA receptors, does the same thing, indicating that indeed, very likely that this is an NMDA receptor mediated response. Now, another important fact here is that NMDA responses per se don't change. So NMDA receptors are blocked. You remove ketamine, they are basically relieved. They are actually happy. They don't actually get altered per se, but that transient block of the NMDA receptor is sufficient to trigger this plasticity. And also we know from the clinic that people get ketamine, although ketamine gets metabolized and cleared off quite rapidly, the antidepressant effect sustained far longer than that. So here is some other data that we have shown where you apply ketamine in the presence of tetrodotoxin. This is kind of a really, really killer experiment because we show that indeed you don't need any activity to trigger this effect. Here, we're stimulating, we're perfusing TTX to block action potentials, no nerve impulses, ketamine is applied, both are washed out, you get potentiation. And TTX by itself doesn't do much. If anything, maybe there's a slight reduction because you cannot wash it out completely. So there's actually robust potentiation you get even in the presence of tetrodotoxin. And you don't get this potentiation when you block protein translation. And you don't get this potentiation when you knock out BDNF. It is BDNF dependent, protein translation dependent, it is activity independent, and depends on NMDA receptors. And then the other thing is this key factor we have shown, EF2 kinase, is required because in a mouse that lacks EF2 kinase, you actually don't get this potentiation. Again, there is also this kind of reverse genetics component of it, like BDNF knockout mice or EF2 kinase knockout mice don't respond. Also behaviorally. So everything I've shown you before actually have also behavioral counterparts. So then we actually pursued this further, and we could show that altering BDNF tract B signaling specifically in the CA1 region of hippocampus is necessary for this effect. So if we knock out BDNF or tract B in only CA1 neurons, we block this potentiation. And in this study we published a couple of years ago, we could show that behaviorally also you can block the behavioral effect when you manipulate this plasticity within hippocampus. So this basically kind of consolidates what I've shown you here before, that indeed there seems to be some sort of a kind signaling transduction elements that like this here occurring in the hippocampus is critical to trigger ketamine's plasticity and also behavioral effects, which I'm not really showing all the behavior associated with this work. And then one other thing before we get into the paper that I'm going to discuss quickly is that we could do, the point is we actually suggesting that spontaneous release of glutamate at resting conditions, absence of activity is critical. So what we have done also, we manipulated two snare molecules that are involved in spontaneous release specifically. And this is a study we published in 2017. And what we could show in this paper is that indeed, when we manipulate these proteins that selectively suppress spontaneous release of glutamate. Now, if we do this on the postsynaptic side, in this case in CA1, we get the plasticity. But if we do it on the presynaptic side, CA3, the input into CA1, we don't get the plasticity. Now, the important aspect of this study, this paper back in 2017 is coming back to the current paper related LTP, long-term potentiation that you trigger by stimulation and activation of NMDA receptors is unaffected. So this already shows that there is this dichotomy between these two plasticities. You can actually molecularly separate them. And here, there's a difference that I would like to highlight. Here, we are manipulating synapses presynaptically, impairing this potentiation. But there, previously, I've shown that if we manipulate synapses postsynaptically, CA1, BDNF, or TrkB, we enhance this, block this potentiation, indicating that indeed this specific form of release is required to trigger the potentiation and activate BDNF, TrkB signaling pathway. So that brings me to the paper that I'm talking about here. We basically tested this quite explicitly. And experiments are actually quite simple. So in this current study, what we have done is that we treated animals with ketamine. We could show that they show rapid antidepressant-like effects. And at that, we actually cut brain slices and then looked at if we could trigger long-term potentiation, the Hebbian plasticity, more associated with learning and memory. And indeed, we could. We could only see some subtle changes, but overall, these animals could actually learn, on the other hand, behaviorally, but in the meantime, they could also elicit, they're susceptible to this form of synaptic plasticity, indicating that ketamine administration didn't affect this plasticity also in vivo. And here we've done another set of experiments. We actually, instead of stressing animals, we actually elicited stress or depression-like behavior, you could call, with administration of corticosterone. So corticosterone administration causes behavioral outputs that look like this anhedonia-type behavior that normally assessed in the lab. That's kind of a correlate for, human depression. Of course, these are not really outstanding. These are not really perfect models, but still they actually help us to work through these signal transduction pathways. And simply what we see is that this corticosterone administration doesn't alter as much baseline synaptic transmission. This is kind of all negative data. And in turn, corticosterone application or administration to the animal also does not affect long-term potentiation. So these animals, I mean, this also, I guess, is, it's not that surprising. You might be stressed, but it's not like your learning and memory is disabled. So that's clearly also can be seen in these slices, still can trigger this long-term potentiation. And this is basically the final experiment under the same condition, either under normal circumstances or after CORT administration. You can trigger ketamine-induced plasticity and then subsequently also can elicit LTP. This comes back to the point I made before, indeed showing that even in the same slice, side by side, with the prior administration of corticosterone or not, ketamine-induced plasticity and long-term plasticity that's associated with Hebbian-type plasticity, learning memory, are independent. So this is pretty much the bottom line of this paper. And I have more slides and more data that I'm happy to share. But I think maybe we should start a more of a conversation that I'm happy to refer to those points. And this paper is interesting in the sense that overall, actually, the findings are negative. But I think it's important negative data. First, I think it totally recapitulates something that is seen out there in the clinic. And plus, importantly, in many aspects of the ketamine research, people confuse long-term potentiation and the ketamine-induced plasticity. And what we try to really highlight in the study is that those two processes are independent. That, I think, is the key thing. There's, of course, they work at the same type of synapses. They both cause potentiation of AMPA receptors, but they work independent of each other. I think that's the key important point. And even some people in the synaptic field tend to confuse these two forms of plasticities. Thank you.
Thank you very much. I wonder if
Right.
anybody
Stop
has.
sharing, or how do we go from here to move on to our like discussion phase, right?
You can just like leave it on.
Okay.
Sometimes, let's say if there is any specific question about your presentation, it's just easier to maybe go back to any graph that you might find that
Yeah.
helpful. And again, for people who maybe is not familiar with the format, you can just like feel free to, raise your hand or like ask a question directly. Or if you're feeling a bit too shy today, you can just like type this in the chat and then we can read it out loud.
Yeah, there's no need to feel shy.
So in the meantime, maybe I can start asking you a question, Dr. Cavallari,
Yeah,
if that's all right.
sure.
So you since from your work you really focus mostly on the hippocampus, but do you
Yeah.
think that this is effect of ketamine also translate to other areas of the brain?
Yes. So clearly there are several brain regions are involved. I think there is good evidence, for instance, prefrontal cortex being involved. And there is a very nice work originating in China suggesting that lateral habenula, that's actually work originally done in Cold Spring Harbor, Robert Molyneux's lab, and now in China, suggesting that lateral habenula plays a role. So the question here is that our work indicates that this type of plasticity in hippocampus is necessary. Now, is it sufficient? That's a whole other aspect. And it is actually, how to say, difficult to sort that out, which is something that we would love to do. I mean, there is, of course, now all these optogenetic methods. But on the other hand, there is, as you can see, some level of subtlety to this plasticity. So just increasing activity overall, for instance, I don't think is a good, let's say, a correlate to do that type of. I mean, I'm giving a long answer to your question because, so yeah, it is. I think hippocampus is a key region involved and necessary. On the other hand, we can definitely cannot exclude. The only thing I would say is that I see some of my colleagues in the field claim this is the region. I think it's hard to see that. I mean, the way that ketamine works in the clinic, what I can see is it works against in several types of depression, amazingly. And not all those the types of depression actually share the same pathophysiology. So therefore, I would expect that this type of plasticity, this kind of mechanisms may work in different brain regions to trigger this effect. And yeah, I'll stop there, but I actually have a couple more comments to make in that regard if during the discussion.
Thank you. I think we have a we have a question in the chat. Tiago, you're happy to ask yourself, or I can read this if you prefer.
Today I'm having lunch, so I'm not in my best picture, so please do it for me.
Do I see the chat? I don't see the chat here.
Oh, you don't, but basically it was like, the comment was like, that's a really dense preclinical data. As I myself am a clinician, help me with take home or take to the clinic key points.
Yeah, so I think the key point is I think I tried to emphasize that. So I think it all comes back to this concept of plasticity that is kind of used over and over again now in the field of neuropsychiatry. So yes, plasticity, synaptic plasticity is important to trigger these beneficial effects of ketamine or maybe also psychedelics and many other compounds. And in the meantime, there are plasticities that are involved for processes like learning and memory, for sure. And what we're trying to say is that there are different types of plasticities. And in this case, what we are talking about, ketamine is triggering a form of plasticity, which we kind of tend to call more of a rapid homeostatic plasticity that is critical for ketamine effect. But this plasticity does not necessarily impact other forms of plasticity, works independent of them. And I think that is actually a good thing. It is a good that you can take ketamine, use ketamine or other, hopefully other drugs like ketamine, and that do not necessarily impair your learning and memory, other cognitive processes. I think that's the bottom line, that these two things inherently work very differently.
So see, we have some raised some raised hand, and then we have another question in the chat. Dr. Cavallari, you can see the chat in the bottom. There's like participant, chat, react. I don't know if you see it. Otherwise, I can.
I don't see it. I don't know
why.
what
Okay.
should I move back from screen sharing? Then
You can try. You can most certainly try.
Okay, let's say stop share. And then I see chat now. Okay, I see the chat.
So you have a question.
So why memantine doesn't give the same effect of ketamine in antidepressant and LTP neuroplasticity on NMDA blocking or memantine? Okay, excellent question. I actually have data on that. So we worked on that. That's actually a very important point. So memantine and ketamine, if NMDA receptors are open and activated the way that, they would be activated during, for instance, long-term potentiation or something like that, and memantine ketamine works the same way. So, and there's very little difference between them. So we looked into this, why memantine doesn't work as a rapidly acting antidepressant. And our proposal is, what we think from the very basic data is that memantine is simply not good at blocking NMDA receptors when they are partially occupied by magnesium under resting conditions. Yes, this is a loaded sentence, but let me explain. So one of the key properties of NMDA receptors that makes them so important is this coincidence detection. And the coincidence detection property of NMDA receptors is encoded by their blockade of magnesium ions under resting membrane potentials at low potentials, and it is unblocked by stimulation. So therefore, not only having glutamate is not sufficient to activate them sufficiently, open up and cause large influx of calcium. So what is critical is that you need to depolarize, means increase the membrane potential, unblock magnesium, and also activate using glutamate. Now, memantine can block that, but memantine is terrible in blocking these receptors that are only partially unblocked under near resting membrane potentials where memantine has to compete with magnesium. That is our explanation. We looked at this explicitly. I can show the data, but that data will actually get really, really dense. But others, actually work on the same question, and they have other observations that I think there is this really comes down to the biophysical properties. So that actually is important, because whenever somebody claims that such and such mechanism is important for ketamine's antidepressant action, I think they should also perform with experiments with memantine, because memantine has not been through all kinds of trials. A lot of people wanted memantine to work, and probably I would have liked memantine to work because presumably it would have less side effects, etc. But memantine doesn't work. But that's a good negative control. So because ketamine is kind of a dirty drug. And now, I'm a chair of pharmacology, and now a lot of pharmacologists actually call it like polypharmacological compound. It used to be a dirty drug, but now it's called polypharmacology. It is true, has several targets, but NMDA receptor is the key, and NMDA receptor biophysics is very important why memantine doesn't work. And we actually published this almost a decade ago now, showed that memantine actually does not activate the signal transduction pathway that I showed you, and mainly because it doesn't work at rest the way the ketamine does. Sorry, long answer, but a very important question.
Rupert, I think you're next in line.
Thanks. Yeah, I saw a patient, 77-year-old patient this morning, and the issue that we discussed was cognitive impairment, which obviously speaks exactly to what you're talking about. And I felt obliged to tell her that in young people, there's no evidence at all that ketamine impairs cognitive function. In fact, if anything, it gets better perhaps because the depression gets better and people can think more clearly.
Yeah.
That was the sort of level of explanation that I'm giving to patients. But there is a trial that was done in over 65, people over the age of 65, which with S-ketamine nasal spray. And I felt obliged to tell her that I think that there was one of the tests about, and it was either simple or choice reaction time, and there may be clinicians on this call that can help me with this, that actually was significantly worse after a prolonged period of S-ketamine, I think after a year of S-ketamine.
Yeah.
I wish I didn't have to tell her that.
Yeah, so that is actually, I think, another very important point. So these are, of course, all these experiments that I've shown you, mostly pinpoint towards an acute application, single dose, in a preclinical setting, or, and then you could trigger this plasticity and how that interacts with long-term, Hebbian plasticity or not. Now, sustained administration of ketamine, which is quite often required to actually see or increase ketamine efficacy.
Or repeated.
Repeated, yeah, it's not sustained, sorry. That is, we actually worked on that. And in the animal models, we could actually show that additional mechanisms come into play when you actually look into the long-term effects of ketamine. Definitely transcriptional mechanisms are important. And that's actually a very important area. So I would not be surprised that repeated administration will start having somewhat, let's say, unpredictable side effects. That is not going to be surprising to me. And that's something we were trying to understand, to be honest. And it's somewhat, it's closer, it's, let's say, we published some of this, but it's more to come to look into this, because the problem is, yes, if patients going to be on ketamine for a long period, how can we alleviate other potential side effects? So that's a concern. That is a concern. But at this point, from a preclinical synaptic perspective, I can only hand wave what might be happening in the long term. So that's yeah, but that's a very important area. If we need to, if ketamine is going to become a routine, it doesn't not only work rapidly, but you should also be administered over time and maintain efficacy over time. And how that's going to be, it's a challenge.
I hope we won't inhibit you from hand waving, actually, because that's part of the fun.
Yeah, it is the fun. It is, you can always, I think the problem with ketamine field, there's so much hand waving that I am really trying not to hand wave. Right. Other fields, I've been involved in other synaptic fields, and I'm happy to hand wave. But ketamine, I feel there's so much stuff out there that, what I love as a very basic synaptic physiologist, that this is a tractable problem. It is a tangible problem. And, we can build, falsifiable hypotheses that people can go and find out if they can make it better. It is not one of these. I think neuropsychiatry has a lot of treatments and ways that people say, hand waving, say, it's complex, but it works. In ketamine, I am very optimistic, maybe naively, that this is actually tractable. Yes, questions?
I think, Kate, if you want to ask your question,
yes.
Should be.
Hi, thank you. I have a bit of a potentially naive question, but I was wondering, with the non-LTP plasticity that you're measuring, if you could elaborate a little bit more about what exactly that is measuring and how that differs, or does it differ from just enhanced excitability?
Yeah, I will. So yeah, that is again, it can easily get dense, but let me explain. So this type of plasticity is being observed, actually has a long history in physiology. So what happens is, if synapses or circuits, when they are not used, this goes back to work of people like Walter Cannon and others back in 50s and 40s. They have studied this in peripheral nervous system. If you denervate muscle, for instance, the muscle becomes oversensitive to acetylcholine. This goes in, very, very fundamental physiological observation. And this is late 1990s. Gina Torrigiano and colleagues at Brandeis actually observed this, such that if they silence neurons for over a period of days, what they have detected is that synaptic strength gets stronger. So it's some sort of synaptic potentiation that occurs in response to prolonged neuronal silencing. So this has become called homeostatic synaptic plasticities. And the way they happen is called synaptic scaling. Now, this is actually an important concept because when you trigger LTP, what you want is you want to strengthen the certain synapses that your pathway is targeting and not others. So you want to be very synapse specific when you do LTP. Now, in this plasticity, you want to regulate the level of each synapse equally, but equally in proportional to their original strength. It's called synaptic scaling. So everything is multiplied by a factor, okay, or divided by a factor. It can go both ways, upscaling, downscaling. In this way, what you're doing is you're adjusting overall strength of synapses across a neuron, okay, but you're not changing their relative strengths to each other. This comes back into exactly the point I'm trying to make. When you do LTP, you want to pick out synapses. I want to strengthen these guys, not the others. In this case, you want to up or down regulate all of them at the same time, but in such that you don't want to disrupt the information encoded in their relative strengths. Does that make sense? Now, the point that the surprising thing that what we see is ketamine is that this type of plasticity, normally thought to be triggered for over like days, can be triggered really fast within an hour or so. So we therefore call this rapid homeostatic plasticity. And the reason that you can trigger this really fast is because you're not only blocking activity, you actually even don't need to block activity. All you need to do is to suppress that spontaneous neurotransmission. That neurotransmission that occurs under resting conditions needs to be blocked. When you suppress that, you get this plasticity and you get it on a faster time scale. And that is because that plasticity, that spontaneous release has this suppressing effect on dendritic protein translation. So when you unblock that, suddenly you get this plasticity and you get it on a faster time scale.
Thank you.
I think we have a couple of questions in the chat.
Yes. So memantine, and then the other one. So are you, as a Genius Hamid, so are you implying that the preloading with magnesium prior to ketamine administration that I have heard some colleagues do may actually slow down the mechanism of ketamine, or does ketamine action function despite magnesium blockade? That's an interesting question, actually. So I know there are, I actually have some former colleagues who've been working on this magnesium impact on NMDA receptor function, and if that is beneficial or detrimental. So to be honest, since we haven't done the experiment, but that's actually suggests an interesting experiment that we may pursue. This is interesting. Increasing magnesium levels and completely taking NMDA receptors, kind of blocking them even stronger, would it actually make ketamine effect more robust or no? I don't know the answer to that. And that's an important point because we published another paper together in Lisa Montegia's group. What we show is that a compound, another endogenous neuromodulator called relin, which is a large glycoprotein in the brain that is important to sustain an NMDA receptor activity, is important for ketamine action. So that, in a way, suggests that maybe if we increase magnesium too much, really suppressing an NMDA receptor activity too much might actually be detrimental. So, but I don't know that. But I know that there is some NMDA receptor activity. Yeah, that's an interesting point, but I don't know the answer. So, so the other next question, Dr. Ahmed Al-Bahari, can we say that other antidepressants would help if LTP with ketamine? So if we can use other antidepressants that adjuvants to ketamine therapy to augment LTP or BDNF production neuroplasticity process. So that is, that's a very clinical question. That's actually a question that I've been asking and trying to find out. People probably try, maybe starting initial ketamine and then continue with SSRIs. That's something question that I would ask you guys more in the clinic, if that is something that actually people do and how the response is. I'm not aware of clinical trial that actually gave a clear answer to that, but I would love to know actually. So that's actually, I'm kind of reversing that question back to you as the clinicians in the field. If that is something I would, there is this, how to say, clearly convergence on BDNF signaling for both types of antidepressants. So I don't know if anybody wants to comment on that.
My sense of this is that you're right about there not being a clinical trial looking at the addition or not of an SSRI to ketamine. But obviously, from a regulatory point of view, with S-ketamine is licensed for use in addition to an SSRI. But there may be others who've got other data. Our clinical impression is that sometimes people do seem to do a bit better if they're on an SSRI as well. But that's just a hand waving, as you say.
I think it's a bit difficult because a lot of those trials in ketamine, right, were initially run on patients who were treatment resistant but drug-free because they really want to establish the effect of ketamine. And then, as Rupert said, they were like the FDA EMA approval trial, the sustain and the transform for esketamine, that is usually an add-on. But I cannot recall myself neither about a trial where like we start somebody on ketamine and then we added the SSRI. It only similar things that I can think is a trial when we gave ketamine and then we added lithium on it.
Oh, that's interesting. So we actually
exactly for that the work that Lisa did on these elongation factor and the GSK3 pathway.
Yeah. So that's actually interesting. Yeah, we have actually studied the lithium effect. I mean, that has an antidepressant effect. I mean, it is coupled with it's more of a mood stabilizer. I know the lingo is somewhat different, but that's actually very interesting. And that is more what's in our hands. Lithium is more of a sustained increase in BDNF, and that actually is given also more chronically. And we think they actually have different effects on synaptic plasticity. So BDNF is actually quite interesting. I think depending on where it's released, where it acts, how long it is there, it all very different effects. In a way, I more and more compare BDNF to glutamate. I mean, no one would claim that glutamate should do the same thing in every context and everywhere. It is a major neurotransmitter, and different circuits work by glutamate, and each circuit, each brain region has different functions, etc. And no one questions that. I think the same is true for BDNF. It's very diverse functions. So there is Silvana Melendez. Is that question? Yep. I think there's a hand there.
Hi.
Hi.
I just wanted to say I thought your your whole presentation was super interesting, looking at like the homeostatic plasticity. And I was just really curious about this difference because I don't know if you've already like mentioned it in your paper, but I was curious to know whether you've looked into the differences in terms of the AMPA receptor subunits and like whether these two different types of plasticities kind of like maybe affect like GLUA1 or GLUA2, for example, and like maybe I'm a bit curious as well about maybe some of the downstream signaling cascades. Maybe like is there a reason for why they are so different and respond differently?
That's actually an excellent question, and that impinges on some of the maybe current debates going on in the field of long-term plasticity. There have been labs who claim that LTP, elicitation of LTP, triggering LTP, actually causes insertion of GLUA1 lacking, GLUA2 lacking AMPA receptors. And those receptors are actually calcium permeable. Those are called calcium permeable AMPA receptors, CPA-AMPA, that people call it. Now, that's been somewhat debated. Let's say I don't like the word controversial anymore, but I think there are contexts that happens or doesn't happen. Now, in the type of homeostatic plasticity that I talked about, in vitro, other labs who study the same thing, they have seen it. They have seen that you can get GLUA2 lacking AMPA receptor insertion. In our case, we don't. Now, on the other hand, we published another paper where we triggered another form of homeostatic plasticity by adding retinoic acid. So retinoic acid is also kind of cuts to the chase and triggers this homeostatic plasticity, okay? Doesn't use EF2 pathway at all, somewhat different. And it causes insertion of GLUA2 lacking AMPA receptors, definitely. But behaviorally, we actually could see similar effects. So in that study, what we kind of proposed is that maybe you need this homeostatic plasticity. No matter how you trigger it, you can actually get a beneficial effect. Of the caveat is, of course, is retinoic acid, is it going to have other effects? Off target is possible. I mean, that's just a proof of principle, just to trigger the same type of plasticity by a different mechanism. And can you see a similar antidepressant-like effect in a, in a mouse model? But the point is that, yes, having GLUA2 lacking receptors there may not be detrimental, but they would confer other properties that would be very interesting to follow up.
Super interesting. Thank you so much. Thank you.
Sorry, if we just like, Leith, if you want to ask your question directly, just to recapitulate. So overall, do you think that this effect on ketamine is more responsible for the rapid effect of ketamine or also for the sustained effect?
Yeah, so what we know so far is that what I described now is more about the rapid effect. Now, the sustained effect requires transcriptional mechanisms. So we published on that, and I don't think people would disagree that we proposed that a transcription regulator, MECP2, MECP2, which is actually, associated with a devastated neurodevelopmental disorder called Rett syndrome, is a key factor there that gets phosphorylated and activates a transcriptional program. That what happens, what's interesting is, what happens is it doesn't necessarily make the plasticity last longer. What it does is it actually renders synapses more susceptible to further stronger plasticity, what I would call a metaplasticity. So that was the interesting part of those observations, is that they kind of nicely overlap with the observation that repeated administration, as been discussed before, is actually better. And repeated administration actually makes the effect better, but it also makes the synapses more receptive to this type of plasticity. So we showed that. That's all published in a paper, I think, like 2021 or 22, something like that. We published that. So that's interesting. Now, therefore, one of the key things that it's important to think about, the rapid effect is important to trigger that downstream effect. So I don't think you can dissociate those. You have to get that rapid effect, at least in our hands now, to trigger the downstream effect. But downstream effect is activation of distinct pathways, not necessarily the maintenance of the plasticity, but more of an elicitation of a form of metaplasticity. So that's our current thinking on that. So I see some questions here. Dexamethasone suppression test, would it be useful? I saw a presentation suggesting cortisol levels were a predictor of response to ketamine. I don't know. I mean, it is at least in our hands, in the clinic, it might be. But in the preclinically at the synapse, as I showed you, we don't see much relationship. But of course, it all depends also on the long term. That might be a different issue. This is more about the acute effect. The other question is also Sava, a paper suggesting that stress before ketamine might prevent it being kicked out of the channel. So it would imply that stress before infusion could prolong the benefit, though this might be, although we do not find it clinically. I had a patient who swore that aspirin improved the effect of ketamine, Rett syndrome and aspirin. So, so the question is, okay, the couples levels here. Can stress, I mean, at this point, we do not see, at least at the synaptic level, an overlap with stress. So ketamine works actually after chronic stress. Now, what's important, what was interesting, there are actually other experts on this, that social defeat stress, that has been a paradigm that's been used preclinically to model, for instance, SSRI effects. Like it takes longer for SSRIs to work, in a social defeat stress, and which is my understanding. I'm not a behaviorist, but it's a brutal paradigm in many respects. But ketamine has not been as efficacious, has not been this robust response to ketamine in that type of paradigm. Whereas for swim test and, kind of more learn helplessness type tests, and those actually work better with ketamine. Now, on the other hand, this plasticities that I showed, as I showed in this paper, and others in our, you don't actually, stress is not that much of a predetermining factor for this type of plasticity. Although I am very well aware that in the patients, in the clinic patients, if they are not depressed, they are actually, normal, their response to ketamine differs from response of patients who are actually depressed. I know that in the clinic, but in this synaptic level, interestingly, we don't detect a huge difference.
There was a question actually. I was just, yes. I'm afraid I'm slightly scrabbling at the back of my mind, but I did see a paper recently about ketamine being kicked out of the channel.
Yeah,
and I was wondering if you can say something about.
There is this trapping of ketamine. I mean, the Highland Hu paper in, I think, was it Nature, or all her papers are very high profile in one of those journals. And what she suggested, trapping is actually kind of triggers more long-term, more sustained effect. So that's not surprising to me. And that was not surprising. And one of the things that actually I see a name, T.D. Gold, it might be Todd Gold.
It is.
And he actually published an influential paper suggesting that ketamine metabolite is an important for actually this more or less say sustained effects of ketamine. So that could be, their proposal was this is ketamine NMDA receptor independent. As he would know quite well, we kind of disagreed with that type of proposal. But on the other hand, overall, the data is quite compelling that it is possible that, ketamine is metabolized, and these metabolites continue to block the receptor and actually kind of make the effect more sustained. So that, I think, is a possibility. So yeah, I don't know if he would like to comment. If that's him, I'm not sure if that's him, but I see the name there. So questions?
You said that you'd be prepared to talk about other mechanisms of action of ketamine, like opiates. Opiate mechanism, I guess, was what you were referring to right at the beginning.
It is. Yeah, I know. It is. There is a lot of issues with. I can go into. One of the things, for instance, earlier work suggested that rapamycin was, or mTOR signaling is critical. So we kind of, partially disagree. Clearly, protein translation regulation is important, but we were not, so convinced that mTOR signaling was as essential as has been proposed. And then I know that John Crystal and colleagues conducted actually a study on that. And if anything, they see the opposite. In a way, the rapamycin treatment actually made the effect somewhat last longer. So that's an interesting point that could be. One of the beauties of the ketamine work, I always say, like the memantine finding, like the repeated administration finding, there is so much clear, unequivocal information coming out of the clinic that actually guides our. I call this more reverse. Actually, I learned I borrowed that term. Lisa uses that, and I love it. It's reverse translation. Actually, clinic is guiding us in many ways. And so the same thing about rapamycin, what is actually doing is quite interesting. Now, the other aspect is opioid. So to me, I think at the synaptic level, when I look at a synaptic level, definitely, anything that manipulates synaptic efficacy will have some impact on these responses. So it can go in many directions. But do I think that, there is a, let's say, integral role for opioid signaling in the response? I am somewhat skeptical, at least from my, experience. This kind of, there's an analogy to this, which I didn't get into. So BDNF track B signaling originally was proposed to be quite integral for LTP, what I mentioned, long-term potentiation. But in our hands, while BDNF track B signaling is quite regulatory for LTP, I don't think is essential. It's not like essential as NMDA receptor is, for instance. So in the same manner, any other manipulation that you may do that manipulates synaptic efficacy one way or another might have effects on this type of plasticity and the outcome of ketamine responses. But I don't think they are necessarily the part of the core pathway. I hope I could make that, that's the clear thing. So people may come up with things. I mean, one of the other things that has been somewhat actually here having the clinicians on the on the call is actually important. Some studies want to desperately tie ketamine action in the short term to monoaminergic signaling. And that's always, to me, at least my reading of the field, is that there's such a large neuropharmacology of monoaminergic transmission that is involved in like classical antidepressants or, you know, schizophrenia, etc., but not much evidence for rapid antidepressant effects so far. So therefore, while in the long-term effects of ketamine might involve definitely maybe some downstream pathways that have involved monoamine signaling, but the acute effect, I sincerely doubt, has an obligatory role for monoaminergic signaling. If, you know, I hope that's clear what I tried to say.
I mean,
why would the NMDA are the NMDA is the pattern of NMDA receptor subtypes different in the lateral habenula to other areas?
That I'm not sure. I mean, there was been overall claims suggesting that NR2B receptors might be required. I don't know the final word on that. I mean, in one hand, that's the NR2B receptors have higher open channel probability. They open longer in response to the same amount of glutamate. So therefore, they might be more susceptible to block by ketamine, and that's not surprising. But on the other hand, as far as I know, the data so far is I like the word equivocal. You know, you can go both ways. But yeah, I'm not sure about now. There, on the other hand, there are in the cortex, there are NMDA receptors that are not as susceptible to magnesium block. So some actually have proposed that NR2C, NR2D receptors might be, you know, responders. That is possible. But on the other hand, I mean, they will create more tonic signaling with NMDA. That's a whole other issue there. There's a lot of interesting biology that's associated with those receptors. But my view in this, your garden variety NMDA receptor is, I think, is sufficient to trigger this pathway. So you don't need any special NMDA receptor to do this. But of course, again, it can effect may be modulated, may be made better or worse by depending on the subunits.
I think we may have time just for one last question. Is in the chat, if you want to read it yourself. Basically,
oh, there is actually a question. That's important. Would you explain why ketamine as NMDA blocking may induce psychosis or mania vulnerable psychotic patients? Yeah, that's an important point. And as my understanding, the history of this field actually started out, John Crystal, as I know, I mean, he actually worked on psychosis. And this was how this whole thing started. And but this is a very low dose of ketamine. And I know higher doses, and actually Lisa published a study on that showing that the effects that we see, I report here, are actually quite dose dependent also preclinically. I think that's an important point. So like a higher dose of ketamine, would it be as efficacious to actually trigger plasticity? That's actually doubtful to me, this type of plasticity. Now, that could also, of course, get into ketamine's more maybe off-target effects. That's the other problem with that type of approach. But initially, what has been proposed is, you know, ketamine or other NMDA receptor blockers might be selectively targeting NMDA receptors on interneurons. And these interneurons, NMDA receptors, might be, you know, blocked such that they are disinhibiting target excitatory neurons. And that could be a way that you may be why you may be triggering psychosis. And because you get this glutamate surge, it's called, because you can see it, I think, using more like microdialysis experiments. And I think you can also detect this with functional MRI type approaches that you can actually detect this, you know, glutamate increase. So to be honest, at that point, that's very well possible. And this may be also I wouldn't contest that something like this might be happening with ketamine administration in there. Some people actually show, even low doses, some sort of, dissociative effects. I doubt that those effects are integral to the antidepressant response. I know that this is a kind of a point of debate, and some may disagree. I think those effects are, I would call it maybe one of those off-target effects that I think we can avoid. But that's a whole other issue. And that's probably triggered by different pathways. And one of the things I can tell you is there's also very well described, although in the data that I showed you, we do not see a presynaptic change, meaning that any change that would increase the amount of glutamate release. In our hands, we don't see that. I cannot fully exclude that something like that might also be happening. But I don't think it is happening. If it is happening, it's not happening in the wave of a surge of glutamate. It's more probably an increase in glutamatergic release probability such that some augmentation, presynaptic augmentation, which might be happening too. So there might be more, prone to triggering psychotic type effects. And that could be an interesting kind of a preclinical question by itself. But at this point, I feel, at least our data and others, what I know, I think these two things are dissociable. Psychotic type, dissociative effects of ketamine and the antidepressant effects.
Well, thank you very much, Dr. Cavallari. I'm afraid to say that at the time of our journal club today is coming to an end. But it was, I feel, a very productive and interesting and thought-provoking discussion. So thank you very much.
I'm very happy to hear that, and thank you for including me in this. Thank you very much.
Great.